In the deepest corners of the universe, hidden within the fabric of reality itself, lie secrets that have haunted humanity for millennia. Secrets written not in stone, not in stars, but in the very code of life itself. Secrets that, when unveiled, would transform our world in ways so profound, so utterly revolutionary, that the course of human civilization would split into before and after. Before we understood, after we knew. These are the moments when a single observation, a chance encounter, a flash of insight, shattered everything we thought we knew about existence. And here's the question that should keep you awake at night. What if every comfort you enjoy, every year added to your life, every miracle of the modern world, traces back to just a handful of these moments? What if everything you are, everything you know, everything you will ever become, exists only because someone somewhere asked a question no one dared to ask before? Picture this: September 28, 1928. A laboratory in London, unremarkable in almost every way. Petri dishes scattered across benches. The smell of bacterial cultures hanging in the stale air. And one man, Alexander Fleming, returning from a Scottish holiday, utterly unprepared for what awaited him. He walks to his workbench. He begins sorting through the dishes he had left before vacation. Dishes containing colonies of Staphylococcus bacteria, the kind that cause boils, sore throats, deadly infections that could turn a simple scratch into a death sentence. But something is wrong. One of the dishes is contaminated. A blob of mold has invaded his carefully cultivated bacteria. Any other scientist might have cursed, tossed it in the waste bin, started over. But Fleming pauses. He looks closer. Around the mold, in a clear zone like a halo, the bacteria have died, completely vanished, as if something from that mold had reached out and killed them. Do you realize what you're looking at when you imagine that moment? You're looking at the instant when humanity gained the power to fight back against infections that had killed billions throughout history. When Fleming noticed that a rare strain of Penicillium notatum had secreted something that inhibited bacterial growth, he didn't just make a scientific observation. He opened a door to a future where a simple cut wouldn't spiral into sepsis, where pneumonia wasn't a death sentence, where mothers didn't have to watch their children die from scarlet fever or diphtheria. Yet here's what haunts me about this story. Fleming later admitted that when he woke that morning, he certainly didn't plan to revolutionize medicine by discovering the world's first antibiotic. It was an accident, a contaminant, a mistake that saved hundreds of millions of lives. Think about that. How many other life-saving discoveries are sitting right now in contaminated petri dishes, in failed experiments, in garbage bins, waiting for someone to notice what shouldn't be there? But the journey of penicillin from that moldy dish to the drug that would save countless soldiers in World War II took 20 years of grueling work, and Fleming wasn't the one who got it there. The scientific community greeted his 1929 publication with yawns and indifference. Fleming found it impossible to isolate large quantities of this precious mold juice. The substance was unstable, difficult to purify, and his efforts stalled. For a decade, nothing happened. The breakthrough languished, and people kept dying from bacterial infections that penicillin could have stopped. Imagine being Fleming during those years, knowing you discovered something important, watching it gather dust while the world remained ignorant of its power. Then in 1939, Howard Florey and Ernst Chain at Oxford University became interested in penicillin. They assembled a team that included Edward Abraham, Norman Heatley, and Margaret Jennings. They developed methods for cultivating the mold, extracting it, purifying it, storing it. They created assays to measure its potency. And in 1941, they had enough to attempt the first human trial. Albert Alexander, a 43-year-old policeman, had developed a life-threatening infection from a scratch in his rose garden. The streptococci and staphylococci had spread to his eyes, his lungs, his shoulder. He was dying. The Oxford team began injecting him with their purified penicillin. After 5 days, he began to recover. His fever dropped. His abscesses began to heal. It was working. It was actually working. But they ran out. They didn't have enough penicillin to complete the treatment. And Alexander died anyway. Can you imagine the frustration? The agony of knowing you had the cure, but not enough of it? The team resorted to desperate measures. Did you know that around 80% of penicillin is excreted unchanged in urine? Dr. Ethel Florey became famous for the Pee Patrol, cycling around Oxford collecting urine from treated patients so they could extract and recycle the penicillin. They were literally recycling urine to save lives. How desperate would you have to be? How certain of your discovery? But World War II changed everything. The U.S. Department of Agriculture set up a lab in Peoria, Illinois, specifically to solve the penicillin production problem. They discovered that adding corn steep liquor, a byproduct of cornstarch production, to the mold broth increased penicillin yield exponentially. They launched a global search for better penicillin-producing mold strains. Soil samples arrived from around the world, but the solution was found at a local market. Mary Hunt, a lab assistant, found a rotting cantaloupe melon. The mold on that melon produced 6 times more penicillin than Fleming's original strain. A rotting cantaloupe from a market in Peoria, Illinois, became the source of most of the world's penicillin production. By 1943, pharmaceutical companies had developed deep fermentation techniques that allowed mass production. American soldiers landing in Normandy on D-Day carried penicillin that saved thousands who would have died from infected wounds. Fleming, Florey, and Chain shared the 1945 Nobel Prize, and the age of antibiotics had begun. But here's something Fleming said in his acceptance speech that should chill you. He warned that overuse of penicillin might lead to bacterial resistance. In 1945, before antibiotics were even widely available, he already saw the seeds of the crisis we face today, where superbugs resistant to multiple antibiotics kill hundreds of thousands each year. He saw it coming. We didn't listen. Now let me take you back further. Back to 1687, to a book that would reshape how we understand reality itself. Philosophiae Naturalis Principia Mathematica, the mathematical principles of natural philosophy. Few people read it. Even fewer understood it. But everyone knew it was a great work. Inside this book, Isaac Newton did something that still seems almost impossible. He explained why an apple falls to the ground and why the moon orbits the Earth with the same theory. He unified terrestrial and celestial mechanics into a single, elegant framework: universal gravitation. The idea that every particle in the universe attracts every other particle with a force proportional to their masses and inversely proportional to the square of the distance between them. But let's talk about how he got there, because the story is stranger than the myth. Yes, the apple incident happened. Newton himself told the story multiple times late in his life. But it wasn't that an apple fell on his head and he suddenly understood gravity. That's a simplification. A fairy tale. What actually happened, according to Newton's own account to multiple colleagues, including the French philosopher Voltaire, was more subtle and more profound. He was sitting in his garden at Woolsthorpe Manor during the plague years of 1665 and 1666, when Cambridge University had temporarily closed. He watched an apple fall straight down from a tree, and he began to wonder, why straight down? Why not sideways? Why not upward? There must be a force pulling it toward the Earth's center. But here's where it gets interesting. Newton didn't stop there. He started thinking, if there's a force pulling the apple down, how far does that force extend? Just to the top of the tree? To the top of mountains? What if it reaches all the way to the moon? And if it reaches the moon, what if the same force that makes the apple fall is the force that keeps the moon in orbit around Earth, that keeps Earth in orbit around the sun, that governs the motion of all celestial bodies? This was revolutionary. For thousands of years, scholars believed that terrestrial physics and celestial physics were fundamentally different. Aristotle taught that earthly objects naturally sought the ground because Earth was their natural place. while celestial objects moved in perfect circles because the heavens were perfect and unchanging. Newton demolished this entire worldview. He showed that the force pulling an apple to the ground and the force keeping planets in their orbits were one and the same. But let me ask you something that Newton himself struggled with. How does gravity actually work? Newton could describe it mathematically with perfect precision. He could predict planetary motions with extraordinary accuracy. But he couldn't explain the mechanism. How does the Earth reach across empty space and pull on the moon? How does the sun grip the planets without touching them? Newton was deeply troubled by this. In his writings, he called the idea of action at a distance absurd, and said that no competent thinker could believe gravity could act where it wasn't. Yet his mathematics worked perfectly. His predictions were flawless. For more than 2 centuries, Newtonian gravity reigned supreme, despite no one understanding how it actually worked. It wasn't until 1915 that Albert Einstein finally explained the mechanism. General Relativity showed that gravity isn't a force at all. It's the curvature of space-time itself. Massive objects bend the fabric of space and time, and other objects follow the curves. Imagine a bowling ball placed on a stretched rubber sheet, creating a depression. Now roll a marble across the sheet. It curves toward the bowling ball, not because of a force, but because it's following the curved geometry of the sheet. That's gravity according to Einstein. And here's something that should blow your mind. Newton's laws of gravity, which we still use to send spacecraft to other planets, to predict eclipses, to understand orbital mechanics, are technically wrong. They're an approximation that works extraordinarily well under most circumstances. Einstein's general relativity is more accurate. But even Einstein's theory might not be the final answer. It doesn't work at quantum scales. It doesn't explain dark matter or dark energy. Somewhere out there, there's likely an even deeper theory waiting to be discovered. But let's return to Newton for a moment, because his achievements extended far beyond gravity. He invented calculus independently of Gottfried Leibniz, though a bitter dispute over priority would poison much of Newton's later life. He revolutionized optics by demonstrating that white light is actually a mixture of all the colors of the rainbow. He designed the first reflecting telescope, using mirrors instead of just lenses to eliminate chromatic aberration. And he formulated the 3 laws of motion that became the foundation of classical mechanics. The first law, an object at rest stays at rest, and an object in motion stays in motion at constant velocity, unless acted upon by a force. The second law, force equals mass times acceleration. The third law, for every action, there is an equal and opposite reaction. These seem simple now because we teach them to children. But think about what they actually mean. They mean that you can predict with mathematical certainty exactly how objects will move if you know the forces acting on them. They mean the universe operates according to laws, not whims. They mean that the same principles that govern a falling apple govern the motions of planets and stars. Newton took the chaos of observed phenomena and revealed the deep order underlying it all. But Newton himself was a strange man, solitary and reclusive when young, vain and vindictive in later years. He was born prematurely in 1643, so small his mother said he could fit in a quart mug. He initially failed to shine as a student at Cambridge. But during those plague years of 1665 to 1666, when he returned to his family farm, he later described this period as the prime of his age for invention. In those 2 years, he developed the foundations of calculus, optics, and gravitation that would make him famous. Now I want to jump forward to 1953, to one of the most famous and most controversial discoveries in the history of science: the structure of DNA. On February 28, 1953, James Watson and Francis Crick determined that DNA is a double helix, a spiral consisting of 2 strands wound around each other. On April 25th, Nature published 3 papers describing DNA structure, one from Watson and Crick at Cambridge, one from Maurice Wilkins and his team at King's College London, and one from Rosalind Franklin and Raymond Gosling, also at King's College. And in 1962, Watson, Crick, and Wilkins shared the Nobel Prize for discovering the secret of life. Notice anyone missing from that list? Rosalind Franklin died of ovarian cancer in 1958 at the age of 37, before the Nobel Prize was awarded. Nobel Prizes aren't given posthumously, but even if she'd lived, would she have received it? For decades, the story was simple. Watson and Crick stole Franklin's data and took credit for her work. Franklin was portrayed as the wronged heroine, the victim of sexism and betrayal in a male-dominated scientific establishment. And there's truth to that portrayal. Watson's 1968 book, The Double Helix, depicted Franklin with shocking sexism, referring to her by the nickname Rosie that she hated, describing her as obstinate and uncooperative, suggesting that if only she would dress better and be more feminine, she might be taken more seriously. It's hard to read now without cringing, but recent scholarship suggests the truth is more complicated. Evidence from letters, lab notebooks, and the recollections of people who were there indicates that Franklin may have been a more willing collaborator than previously believed. She shared data with Watson, Crick, and Wilkins. She knew they were working on the structure. The famous photograph 51, the X-ray diffraction image that Watson said made his mouth fall open and his pulse race when he saw it, may not have been shown to him without Franklin's knowledge. She may have shared it, or at least known it was being shared. So what actually happened? Let's start at the beginning. Franklin arrived at King's College London in 1951 to work on DNA using X-ray crystallography, a technique she had mastered during her postdoctoral work in Paris. She was brilliant, meticulous, demanding of herself and others. She spent the first 8 months designing and assembling a specialized tilting micro camera with her PhD student Raymond Gosling. She developed methods to control humidity carefully, understanding that DNA existed in 2 forms, a crystalline A form when dry and a wetter B form. In May 1952, she suspended a DNA fiber as thin as a strand of hair and bombarded it with X-rays for 100 hours under carefully controlled humidity. The result was photograph 51, showing the B form of DNA with unmistakable evidence of a helical structure. But here's what often gets forgotten. Franklin was a crystallographer. She was trained to study the A form, the crystalline form. That's what crystallographers do. They study crystals. The B form was Messier, less crystalline, harder to analyze with the mathematical tools she'd been trained to use. She was working on both forms, but focused more on the A form because that's where her expertise lay. Meanwhile, at Cambridge, Watson and Crick were building physical models, trying different configurations, seeing what fit the available data. They were using a different approach. Not X-ray crystallography, but model building informed by chemistry, by stereochemistry, by the rules of how atoms bond and how molecules fold. In January 1953, Watson attended a seminar where Franklin presented data on both the A and B forms. He also saw a report written by Franklin that had been shared with the research committee overseeing King's College's work. This report contained key measurements and conclusions from her X-ray studies. And Watson saw photograph 51, either shown to him by Wilkins or with Franklin's knowledge. The exact circumstances remain debated. What's not debated is what happened next. Armed with this data, Watson and Crick built their model. 2 antiparallel strands running in opposite directions. The sugar phosphate backbone on the outside. The bases pointing inward. Adenine pairing with thymine. Guanine pairing with cytosine. A double helix structure that immediately explained how DNA could replicate. Each strand could serve as a template for a new complementary strand. On February 28th, they had the structure worked out. They invited Wilkins to see it. Then Franklin came to see it. According to accounts, she looked at the model and said it was very nice, or something to that effect. She didn't object. She didn't claim they'd stolen her data. In fact, in the coming months, Franklin and Crick became friends. She visited the Cricks regularly. She even stayed with them. This doesn't sound like someone who felt betrayed. But then why did the story become about theft? Partly because of Watson's book. Partly because Franklin died before she could tell her own story. Partly because the sexism was real even if the theft wasn't. Franklin faced obstacles her male colleagues didn't. At King's College, she wasn't allowed to eat in the senior common room because she was a woman. She was expected to defer to male colleagues. Her brilliance and independence were seen as threatening. And there's something else that gets forgotten in the focus on DNA. Franklin's later work on viruses was equally groundbreaking. After leaving King's College in 1953, she moved to Birkbeck College and began studying the structure of viruses, particularly tobacco mosaic virus. She made fundamental discoveries about how RNA is packed within the protein shells of viruses. She was widely recognized as being at the top of her field. She published 17 papers on viruses between 1953 and 1958, continuing to work until days before her death from ovarian cancer, possibly caused by her extensive exposure to X-rays. She was 37 years old. Here's what April 2023 research concluded. Franklin should be recognized not as a victim, not as a wronged heroine, but as an equal contributor to the discovery of DNA structure. She independently grasped how DNA's structure could specify proteins. She provided crucial data without which Watson and Crick later admitted their model would have been most unlikely, if not impossible. And she verified their result with her own continued research. The discovery of DNA structure was a collaborative effort involving 4 brilliant scientists, not a race won by 2 men who stole from a woman. Ask yourself this. How many other women in science have had their contributions minimized, their names forgotten, their work attributed to male colleagues? How many discoveries throughout history carry the wrong names? But let's talk about what the DNA discovery actually means. Why does it matter? Because DNA is the molecule of heredity. Every living thing on Earth uses DNA to store genetic information. The sequence of bases along the DNA molecule, the order of as TS, GS, and CS, contains the instructions for building and operating an organism. The double helix structure immediately suggested how DNA replicates. Unzip the 2 strands, use each as a template, and you get 2 identical double helices. This explained how genetic information passes from parent to offspring, from cell to cell. Understanding DNA structure launched the entire field of molecular biology. It made possible genetic engineering, DNA sequencing, PCR, cloning, gene therapy, personalized medicine, DNA forensics, evolutionary biology based on genetic comparison, and thousands of other applications. In 1990, scientists began the Human Genome Project, an international collaboration to sequence all 3 billion base pairs of human DNA. It was the largest collaborative biological project in history. By 2003, they had a complete sequence. Think about what this means. We can now read the genetic blueprint of our own species. We can identify which genes are responsible for which traits and diseases. We can compare human DNA to Neanderthal DNA and see that modern non-African populations carry 1 to 2% Neanderthal genes. We can trace human migrations across continents. We can develop targeted therapies based on an individual's genetic makeup. We can use CRISPR to edit genes with precision. We're rewriting the code of life itself. Which brings us to CRISPR, one of the most revolutionary discoveries of the 21st century. CRISPR-Cas9, the genetic scissors that can cut and edit DNA with unprecedented precision. Discovered by Jennifer Doudna and Emmanuelle Charpentier, earning them the 2020 Nobel Prize in Chemistry. But like so many great discoveries, this one came from asking questions about something that seemed unrelated to human health. How do bacteria fight off viruses? Bacteria face constant viral attacks. Viruses inject their DNA into bacteria, hijack the bacterial machinery, and produce more viruses until the bacterial cell bursts. But bacteria have an immune system called CRISPR. clustered regularly interspaced short palindromic repeats. When a virus attacks, the bacterium captures a piece of the viral DNA and stores it in its own genome, like keeping a wanted poster. If that virus attacks again, the bacterium recognizes it, produces RNA that matches the viral DNA sequence, and uses a protein called Cas9 to cut the viral DNA, destroying it. Emmanuelle Charpentier was studying Streptococcus pyogenes, one of the most harmful bacteria to humans. In 2011, she discovered a previously unknown molecule called tracker RNA that was part of the CRISPR system. That same year, she began collaborating with Jennifer Doudna, who had vast expertise in RNA biology. Together, they figured out how the system worked. The RNA guides the Cas9 protein to the target DNA sequence, and Cas9 cuts both strands of the DNA. But here's where it becomes revolutionary. Charpentier and Doudna realized they could reprogram the system. They could design synthetic guide RNAs that would direct Cas9 to cut any DNA sequence they wanted. They published their groundbreaking paper in Science in 2012. By the end of that year, 7 papers had already been submitted using CRISPR-Cas9 to edit genomes in human cells, fungi, and zebrafish. Can you grasp how fast this spread? A technique published in mid-2012 was being used worldwide by the end of the year. Why? Because it's incredibly simple compared to previous gene editing methods. Cheaper, faster, more accurate, more versatile. You design a guide RNA to match the sequence you want to edit. The guide RNA binds to Cas9. You deliver this into cells. The guide RNA finds the matching DNA sequence. Cas9 cuts it. Then the cell's natural repair mechanisms either disable the gene or, if you provide a template, insert new DNA. You can delete genes, add genes, fix mutations, turn genes on or off, all with a precision that was science fiction a decade ago. The applications are staggering. In medicine, scientists are using CRISPR to develop treatments for sickle cell disease, beta thalassemia, genetic forms of blindness, muscular dystrophy, cystic fibrosis, Huntington's disease, and certain cancers. The first CRISPR therapy, Casgevy, was approved in 2023 to treat sickle cell disease. Patients who had suffered from painful crises their entire lives, whose red blood cells were deformed by a single mutation in a single gene, are now essentially cured. Jennifer Doudna met Victoria Gray, the first patient treated with Casgevy, and described the experience as the moment the reality of her discovery's impact truly hit her. This is real. This is happening now. In agriculture, CRISPR is being used to develop crops resistant to drought, pests, and diseases. Crops that need less fertilizer, less water, less pesticide. Tomatoes that produce more lycopene. Wheat resistant to powdery mildew. Rice that can tolerate flooding or salt water. Mushrooms that don't brown. Pigs resistant to viral diseases. The potential to feed more people with less environmental impact. But CRISPR also raises profound ethical questions. If we can edit human embryos, should we? If we can eliminate genetic diseases, should we also enhance intelligence, athletic ability, appearance? Who decides? Who has access? In 2018, Chinese researcher He Jiankui announced he had used CRISPR to edit human embryos that were then implanted and resulted in the birth of twin girls. He claimed he made them resistant to HIV. The scientific community was outraged. He had crossed a line. He had created heritable changes in humans without adequate safety testing, without informed consent, without following ethical guidelines. He was sentenced to 3 years in prison. Doudna herself has been a leader in calling for careful consideration of these issues. She organized a conference in Napa Valley in 2015 to discuss the scientific, medical, legal, and ethical implications of genome editing. The consensus was to strongly discourage work on heritable changes in human embryos until the ethical implications could be thoroughly discussed. Because here's the thing. CRISPR is so powerful, so accessible, that we can do things before we figured out whether we should. A high school student with basic equipment could theoretically edit genes. Garage biologists are experimenting with CRISPR. This isn't like nuclear technology that requires massive infrastructure. This is democratized, distributed power over the code of life. What could possibly go wrong? What safeguards do we need? How do we ensure it's used wisely? And yet, think about the good it could do. Imagine a world without genetic diseases. Without sickle cell anemia, cystic fibrosis, Huntington's disease, muscular dystrophy. Imagine crops that could grow in deserts, that could pull nitrogen from the air, that could feed billions more people. Imagine bringing back extinct species by editing the genomes of their closest relatives. Mammoths, passenger pigeons, Tasmanian tigers. Imagine new materials, new medicines, new forms of life designed to solve problems we can't solve now. We're at the threshold of capabilities that our ancestors would have called godlike. The question is whether we have the wisdom to use them well. Let me take you to another threshold moment, another accidental discovery that changed everything. The cosmic microwave background radiation. In 1964, Arno Penzias and Robert Wilson were working with a large horn antenna at Bell Labs in New Jersey, trying to do radio astronomy. But they kept detecting persistent background noise. No matter where they pointed the antenna, no matter what time of day or year, there was this constant hiss like static. They checked everything. They cleaned pigeon droppings out of the antenna. They eliminated every possible source of interference. The noise remained. It was coming from everywhere in the sky equally. It never changed. Frustrated, they contacted a physicist at Princeton, Robert Dick, who immediately understood what they'd found. This wasn't noise. This was the afterglow of the Big Bang. This was the oldest light in the universe, stretched from visible wavelengths into microwaves by 14 billion years of cosmic expansion. This was direct evidence that the universe began in a hot, dense state and has been expanding and cooling ever since. Penzias and Wilson won the Nobel Prize in 1978. They'd accidentally confirmed one of the most important predictions of Big Bang cosmology. But the cosmic microwave background is more than just evidence for the Big Bang. It's a baby picture of the universe. When the universe was 380,000 years old, about 0.2% of its current age, it cooled enough for atoms to form. Before this, space was filled with a hot plasma of electrons and protons that scattered light in every direction. But once atoms formed, light could travel freely. The cosmic microwave background is that first light, finally free to travel through space, carrying information about what the universe was like at that moment. Later satellites, particularly the Cosmic Background Explorer, COBE, in 1989, the Wilkinson Microwave Anisotropy Probe, WMAP, in 2001, and the Planck satellite in 2009 mapped the cosmic microwave background in exquisite detail. And what they found was astonishing. Tiny temperature variations, differences of 1 part in 100,000. These tiny ripples represent slightly denser and slightly less dense regions in the early universe. And those denser regions, they're the seeds of all the structure we see today. Galaxies, galaxy clusters, superclusters, the entire cosmic web of matter, all grew from those tiny quantum fluctuations in the first fraction of a second after the Big Bang, amplified by inflation and shaped by gravity over billions of years. Think about that. Every star, every planet, every atom in your body traces back to quantum fluctuations in the first instant of the universe. The pattern of galaxies across the sky was written in the cosmic microwave background when the universe was 380,000 years old. We can measure it. We can map it. We can test theories of the universe's origin against it with incredible precision. We know the universe is 13.8 billion years old. Not 13 or 14, but 13.8. We know it's flat. We know it's made of roughly 5% ordinary matter, 27% dark matter, and 68% dark energy. We know all this because we can see the baby picture of the universe in the cosmic microwave background. But here's what haunts me about this discovery. Penzias and Wilson almost missed it. They thought it was noise, pigeon droppings, interference. They were trying to eliminate it, not understand it. How many other discoveries are sitting in our data right now, dismissed as noise, as error, as something to filter out? How many patterns are we blind to because we're looking for something else? Science progresses not just by answering questions, but by asking new ones, by noticing what doesn't fit, by investigating anomalies instead of dismissing them. Consider Einstein's General Relativity again. When he published it in 1915, it made a prediction so bizarre that Einstein himself found it hard to believe. Gravitational waves. The theory predicted that accelerating massive objects would create ripples in space-time itself, waves that travel at the speed of light, stretching and squeezing space as they pass. For decades, this was theoretical, impossible to detect. The effect is so tiny. A gravitational wave from two colliding black holes would stretch the distance from Earth to the Sun by less than the width of an atom. How do you measure something that small? In the 1960s and 70s, Joseph Weber claimed to have detected gravitational waves with aluminum cylinders that should ring when a wave passed. But his results couldn't be replicated. It was wrong. Or at least, if there were gravitational waves, they were too weak for his detectors. So scientists built LIGO, the Laser Interferometer Gravitational Wave Observatory. Two facilities, one in Louisiana and one in Washington State, each with two 4-kilometer-long arms arranged in an L shape. A laser beam splits, travels down each arm, bounces off mirrors, and recombines. If a gravitational wave passes through, it stretches space in one direction and compresses it in the other, changing the path length of the laser beams by an amount smaller than the width of a proton. To detect this, LIGO had to be the most sensitive instrument ever built, capable of measuring changes smaller than 1/10,000th the diameter of a proton. And it worked. On September 14, 2015, both LIGO detectors, 3,000 kilometers apart, detected the same signal within milliseconds of each other. A gravitational wave from two black holes that had collided 1.3 billion light-years away. Do you understand what this means? We can now observe the universe not just with electromagnetic waves, light, radio waves, but with gravitational waves. We can detect events that produce no light at all, colliding black holes, merging neutron stars. We can see the universe in an entirely new way. It's like we've been watching a movie with no sound our entire history, and suddenly we can hear. We're discovering things we never knew existed. On August 17, 2017, LIGO and its European counterpart Virgo detected gravitational waves from two neutron stars colliding. And for the first time, telescopes around the world simultaneously observed the light from the same event. They saw gamma rays, X-rays, ultraviolet, optical, infrared, radio waves. This was the birth of multi-messenger astronomy. We're combining information from gravitational waves and light to understand cosmic events in ways previously impossible. And in 2019, we saw something even more impossible. The first image of a black hole. Not an artist's impression, but an actual photograph of the shadow of a black hole, the point where light can't escape, surrounded by a glowing accretion disk of superheated matter spiraling inward. The black hole is in the galaxy Messier 87, 55 million light-years away, with a mass 6.5 billion times that of the Sun. To photograph it, scientists used the Event Horizon Telescope, a network of 8 radio telescopes around the world, synchronized with atomic clocks, working together as a single Earth-sized virtual telescope. They collected petabytes of data. They developed new algorithms to process it. And they produced an image that matched Einstein's predictions from 100 years earlier with stunning accuracy. Think about the journey from Einstein's scribbling equations in 1915 to an actual photograph of a black hole in 2019. Think about the technology, the international cooperation, the brilliant minds, the decades of work. We've gone from Newton's laws describing how objects move to Einstein's curved spacetime to detecting gravitational waves to photographing black holes. Each discovery builds on what came before. Each opens new questions. And we're still just beginning. We don't know what dark matter is. We don't know what dark energy is. Together they make up 95% of the universe's mass energy, and we have no idea what they are. We have theories, candidates, experiments searching for answers. But we don't know. We don't know if our universe is the only one or part of a multiverse. We don't know if space and time are fundamental or emergent. We don't know what happened before the Big Bang or if that question even makes sense. But let's return to Earth, to discoveries that changed daily life in ways even more immediate. Electricity. Can you imagine a world without it? No lights, no refrigeration, no computers, no internet, no phones, no electric motors, no modern medicine, no nothing. For most of human history, people lived in that world. Lightning was a mysterious phenomenon, feared and worshipped. Static electricity was a curious party trick. But in the 18th and 19th centuries, scientists began to understand electricity as a fundamental force of nature. Benjamin Franklin flew his famous kite in a thunderstorm in 1752, proving that lightning was electrical. Luigi Galvani noticed in 1780 that frog legs twitched when touched with metal instruments, leading to the study of bioelectricity. Alessandro Volta invented the battery in 1800, the first reliable source of continuous electric current. Michael Faraday discovered electromagnetic induction in 1831. Moving a magnet through a coil of wire generates electric current. This became the basis for electric generators and motors, the technology that powers our entire civilization. James Clerk Maxwell unified electricity and magnetism into a single theory in 1865, showing that light itself is an electromagnetic wave. Thomas Edison and Nikola Tesla battled over whether electrical power should be transmitted as direct current or alternating current, with Tesla's AC ultimately winning. By the early 20th century, electric grids were being built, power stations were generating electricity, and the modern world was being born. But here's what's fascinating. Understanding electricity also led to understanding the brain and nervous system. In the early 20th century, scientists discovered that neurons communicate through electrical impulses called action potentials. The brain is an electrical organ. Your thoughts, your memories, your consciousness, all emerge from patterns of electrical activity in billions of neurons. Depression, epilepsy, Parkinson's disease, all involve abnormal electrical activity in the brain. Deep brain stimulation, using electrical pulses to treat neurological conditions, is based on this understanding. The discovery of electricity didn't just give us electric lights. It revealed how our own minds work. And it goes further. Quantum mechanics, the theory that describes the behavior of matter at the smallest scales, emerged from trying to understand how atoms emit and absorb light. Max Planck proposed in 1900 that energy comes in discrete packets called quanta. Einstein explained the photoelectric effect in 1905 by showing that light itself comes in particles called photons. Niels Bohr developed a model of the atom in 1913 where electrons orbit in specific energy levels. Werner Heisenberg and Erwin Schrödinger developed the full mathematical framework of quantum mechanics in the 1920s. And everything changed. Quantum mechanics is strange. Particles can be in multiple states simultaneously until measured. They can be entangled, so measuring one instantly affects the other even across vast distances. They tunnel through barriers they shouldn't be able to cross. They exhibit wave-particle duality, behaving as waves in some experiments and particles in others. The theory violates our intuitions about how reality should work. And yet it's the most precisely tested theory in the history of science. Its predictions have been confirmed to more than 10 decimal places. Every piece of modern electronics depends on it. Transistors, semiconductors, lasers, LEDs, computer chips, all rely on quantum mechanical effects. But quantum mechanics also led to one of the strangest discoveries of all: quantum entanglement, which Einstein called spooky action at a distance. When two particles are entangled, measuring the state of one instantly determines the state of the other, regardless of how far apart they are. This doesn't violate the speed of light because you can't use it to send information faster than light. But it does mean that the universe is fundamentally non-local. Things that are far apart can be correlated in ways that seem to defy our notions of space and separation. And now we're building quantum computers that use entanglement and superposition to perform certain calculations exponentially faster than classical computers. We're developing quantum cryptography that's theoretically unbreakable. We're exploring quantum teleportation, quantum sensing, quantum everything. We're on the verge of a second quantum revolution. Ask yourself, what is reality really made of? At the deepest level, what are particles? Are they strings vibrating in 11 dimensions, as string theory suggests? Are they excitations in quantum fields, as quantum field theory describes? Are they information, as some interpretations of quantum mechanics imply? We genuinely don't know. We have theories that work, that make predictions we can test. But the ultimate nature of reality remains mysterious. And maybe that's okay. Maybe the joy of science isn't having all the answers, but the process of discovery itself, the endless quest to understand. Let me tell you about another discovery that seems simple but revolutionized biology: the microscope. Before the microscope, people had no idea that cells existed, that bacteria and other microorganisms existed, that life operates at scales invisible to the naked eye. When Antony van Leeuwenhoek built his microscopes in the 1670s and looked at pond water, he saw what he called animalcules, tiny creatures swimming around. He was the first human to see bacteria, to see protozoa, to see red blood cells and sperm cells. He opened an entirely new world. Robert Hooke published Micrographia in 1665, filled with detailed drawings of what he saw through his microscope. The compound eye of a fly, the structure of cork, which he described as made of tiny boxes he called cells. That word, cells, now fundamental to biology, came from Hooke's observation that cork looked like the small rooms that monks lived in. In the 19th century, scientists realized that all living things are made of cells, that cells come from other cells, that the cell is the fundamental unit of life. This cell theory became one of the foundational principles of biology. And as microscopes improved, we saw more. We saw chromosomes inside cell nuclei. We saw them duplicate and separate during cell division. We saw bacteria and protozoa in exquisite detail. We saw viruses with electron microscopes. We saw the structure of proteins and DNA with X-ray crystallography and cryo-electron microscopy. Every improvement in our ability to see the very small has led to new discoveries, new understanding. And now we have microscopes that can see individual atoms, that can watch chemical reactions in real time, that can image living cells without killing them. We can tag specific proteins with fluorescent markers and watch them move inside living cells. We can see neurons firing in transparent zebrafish brains. We can map the connections of every neuron in a worm's nervous system. We're building complete maps of how brains are wired, cell by cell, synapse by synapse. But here's the question that haunts me: what are we still not seeing? What exists at scales or in forms we haven't figured out how to observe yet? Dark matter and dark energy don't interact with light, so we can't see them directly. We only know they exist because of their gravitational effects. What else might be out there, invisible to us, fundamental to reality, yet completely unknown because we haven't developed the right tools or asked the right questions? How much of reality is still hidden from us? Think about germ theory. For thousands of years, people died from infections and had no idea why. They believed in miasmas, bad air, divine punishment, imbalanced humors. When Ignaz Semmelweis noticed in 1847 that doctors who washed their hands between performing autopsies and delivering babies had lower rates of childbed fever deaths, he tried to convince his colleagues to wash their hands. They mocked him. The idea that invisible tiny creatures on their hands could cause disease seemed absurd. Semmelweis died in an asylum, his ideas rejected by the medical establishment. It wasn't until the 1860s and 70s that Louis Pasteur and Robert Koch proved that microorganisms cause disease. Pasteur showed that fermentation and spoilage were caused by microorganisms, not spontaneous generation. Koch identified specific bacteria that caused anthrax, tuberculosis, and cholera. They established the germ theory of disease, one of the most important discoveries in the history of medicine. Think about how many lives this saved. Before germ theory, surgeons operated with unwashed hands in street clothes. Hospitals were death traps where more people died from hospital-acquired infections than from their original ailments. After germ theory, we developed antisepsis, sterilization, hand washing, antibiotics, vaccines. We went from life expectancies in the 30s and 40s to life expectancies in the 70s and 80s. We eradicated smallpox, a disease that killed hundreds of millions throughout history. We nearly eradicated polio. We turned deadly infections into minor inconveniences. All because we discovered that tiny microorganisms we can't see with our naked eyes can kill us, and that we can fight them if we know they're there. But germ theory also revealed something profound. We're not alone in our own bodies. We each carry trillions of microorganisms, bacteria, archaea, fungi, viruses, collectively called our microbiome. They outnumber our own cells. They live in our gut, on our skin, in our mouth, in every nook and cranny. And they're not just passengers. They help us digest food, produce vitamins, train our immune system, fight off pathogens, even influence our mood and behavior through the gut-brain axis. We're not individuals. We're ecosystems. We're walking, talking habitats for countless other forms of life. And they're part of us, as essential to our health as our own organs. What's more, we now know that mitochondria, the powerhouses of our cells that produce energy, were once free-living bacteria that were engulfed by our ancestors billions of years ago and became permanent residents. The same with chloroplasts in plant cells. Endosymbiosis, the idea that complex cells arose from simpler cells living inside each other, revolutionized our understanding of evolution. We're all chimeras, combinations of multiple lineages merged together over billions of years. Life doesn't fit into neat categories. It's messy, interconnected, constantly merging and diverging and cooperating and competing. And that brings us to evolution, perhaps the most important idea in biology. Charles Darwin published On the Origin of Species in 1859, proposing that all species descended from common ancestors through a process of natural selection. Individuals vary. Some variations make individuals better suited to their environment. Those individuals survive and reproduce more, passing on their traits. Over time, populations change. Over enough time, new species arise. It seems simple, but it explains so much. Why do organisms have traits that seem designed for their environments? Not because of a designer, but because poorly suited individuals died without reproducing. Why are there so many species? Because populations separated and evolved in different directions. Why do similarities exist between different species? Because they share common ancestors. Darwin didn't know about genes or DNA. He didn't know the mechanism of inheritance. That came from Gregor Mendel, who experimented with pea plants in the 1860s and discovered the basic laws of heredity. Traits are inherited in discrete units, later called genes. Some genes are dominant, some recessive. Offspring inherit one gene from each parent. Mendel's work was largely ignored during his lifetime and rediscovered in 1900, around the same time that chromosomes were being observed under microscopes. Scientists realized that genes were located on chromosomes. They worked out how traits are inherited, how mutations arise, how populations evolve. By combining Mendel's genetics with Darwin's natural selection, they created the modern synthesis of evolutionary biology in the early 20th century. And when the structure of DNA was discovered in 1953, the mechanism of heredity became clear at the molecular level. DNA sequences are the genes. Mutations are changes in those sequences. Evolution is the change in DNA sequences in populations over time. We could trace evolutionary relationships by comparing DNA sequences. We could build family trees of life going back billions of years. We could watch evolution happening in real time in bacteria evolving antibiotic resistance, in viruses evolving to evade our immune systems. Evolution wasn't just a theory. It was an observed fact, happening all around us constantly. But here's what amazes me. Evolution has no goal, no direction, no purpose. It's not progress toward anything. It's simply change driven by differential survival and reproduction. And yet it produced everything. The human eye, with its lens and retina and optic nerve. The brain, with its 100 billion neurons and 100 trillion synapses. The immune system, with its ability to recognize and respond to virtually any pathogen. The heart, the lungs, the liver, every organ, every cell, every molecule, all products of blind variation and selection over billions of years. When you really think about it, it seems almost impossible. And yet the evidence is overwhelming. The fossil record showing gradual changes over time. The geographic distribution of species. The existence of vestigial organs and atavisms. The molecular evidence of shared ancestry. The observation of evolution in action. It all fits together into a coherent picture. And evolution has practical applications. Understanding how bacteria evolve resistance to antibiotics helps us develop strategies to slow resistance. Understanding how viruses evolve helps us develop vaccines and predict flu strains. Understanding how cancer evolves helps us design treatments that prevent resistance. Evolutionary medicine is revealing why our bodies are the way they are, why we're susceptible to certain diseases, how we can better maintain health. We're using evolution as a tool, running evolutionary algorithms on computers to solve optimization problems, to design molecules and machines and strategies. Evolution isn't just how we got here. It's a fundamental principle we can harness. Now let me shift gears and talk about something that might seem unrelated but isn't: the Green Revolution. In the 1960s, Norman Borlaug and others developed high-yielding varieties of wheat and rice that could produce vastly more food per acre than traditional varieties. They introduced these varieties to countries facing famine, particularly in Asia. They taught farmers new techniques: irrigation, fertilization, pest control. And it worked. India, which had been importing grain to avoid mass starvation, became self-sufficient in food production. Pakistan, the Philippines, other countries followed. Borlaug is estimated to have saved over 1 billion people from starvation. 1 billion. Think about that number. He won the Nobel Peace Prize in 1970. But the Green Revolution wasn't without costs. It required large amounts of fertilizer, pesticides, and water. It favored large farms over small ones. It reduced genetic diversity by replacing many traditional varieties with a few high-yielding ones. It changed societies, driving migration from rural to urban areas, changing cultures and ways of life that had existed for millennia. And now we face new challenges. Climate change affecting weather patterns, water scarcity, soil degradation, the need to feed 10 billion people by mid-century while reducing agriculture's environmental footprint. We need a second green revolution, one that's sustainable, one that works with nature instead of against it. And this is where CRISPR and other modern technologies come in. We can develop crops that need less water, that resist drought and floods, that fix their own nitrogen from the air instead of requiring fertilizer, that resist pests and diseases without pesticides. We can bring back genetic diversity by editing multiple varieties instead of relying on just a few. We can create vertical farms in cities, reducing transportation costs and land use. We can grow meat in labs from cell cultures, eliminating the need to raise and slaughter billions of animals. The technology exists. The question is whether we'll use it wisely, whether we'll ensure everyone benefits, whether we'll avoid the mistakes of the past. But let me tell you about a discovery that's more recent and potentially even more transformative: fusion energy. For decades, scientists have been trying to recreate the reaction that powers the sun, fusing hydrogen atoms together to form helium, releasing enormous amounts of energy in the process. The appeal is obvious. Hydrogen is abundant. The reaction produces no greenhouse gases. The waste products are minimal and much less radioactive than fission waste. A single kilogram of fusion fuel could produce the same energy as 10 million kilograms of fossil fuel. It's clean, safe, nearly limitless energy. The problem has always been that fusion requires temperatures of millions of degrees, pressures found only in stars, and confinement systems to keep the plasma from touching anything and cooling down. For years, the joke was that fusion was 30 years away and always would be. But in December 2022, scientists at the National Ignition Facility achieved fusion ignition. They got more energy out of a fusion reaction than they put into it. It was the first time in history that had been accomplished in a laboratory. The margin was small, and it's not yet practical for power generation. But it proved the concept works. It showed that fusion energy is possible. And now multiple companies and research groups are racing to build practical fusion reactors. Some use magnetic confinement, using powerful magnets to hold the plasma in a donut-shaped chamber. Others use inertial confinement, using lasers to compress fuel pellets. Some are trying entirely new approaches. If we can get fusion working at scale, it changes everything. Unlimited clean energy means we can desalinate seawater to solve water scarcity. We can synthesize carbon-neutral fuels for aviation and shipping. We can power carbon capture to remove greenhouse gases from the atmosphere. We can run energy-intensive industries without emissions. We can provide electricity to everyone on Earth without destroying the planet. The climate crisis becomes solvable. And it's not just about Earth. Fusion could power spacecraft for interplanetary travel, making Mars colonization feasible. It could power future space habitats. It could enable us to become a spacefaring civilization. But even if fusion doesn't work out, we have other options. Solar and wind power have become cheaper than fossil fuels in many places. Battery technology is improving, making energy storage practical. We're developing smart grids that balance supply and demand. We're electrifying transportation. We're improving energy efficiency. The transition to sustainable energy is happening. The question is whether it's happening fast enough to avoid the worst effects of climate change. And that depends on political will, on investment, on individuals and societies making different choices. Speaking of climate change, let me talk about a discovery that revealed it's happening: the Keeling curve. In 1958, Charles David Keeling began measuring atmospheric carbon dioxide concentrations at Mauna Loa Observatory in Hawaii. Every day, he took measurements. And he found something remarkable. Carbon dioxide levels fluctuate seasonally, dropping during northern hemisphere summer when plants are growing and absorbing CO2, rising during winter when plants die and decompose. But overlaying this seasonal cycle was a steady upward trend. When he started, atmospheric CO2 was around 315 parts per million. Now it's over 420. That steady rise, that inexorable curve climbing year after year, is the Keeling curve. And it's direct evidence that human activities, primarily burning fossil fuels, are increasing atmospheric carbon dioxide concentrations. This might seem obvious now, but it wasn't at the time. People didn't know if the oceans or biosphere would absorb the extra CO2. They didn't know if human emissions were large enough to matter on a global scale. The Keeling curve provided the evidence. And combined with physics showing that CO2 traps heat, with ice core data showing past correlations between CO2 and temperature, with computer models predicting future warming, with observations of rising temperatures, melting ice, rising seas, changing weather patterns, the picture became clear. We're changing the climate. It's happening. It's real. It's dangerous. And this brings up something crucial about science. It's not about certainty. It's about evidence and probability. We can never be absolutely certain about anything. There's always some possibility we're wrong, that we've missed something, that a new discovery will overturn our understanding. But we make decisions based on the best available evidence, knowing that more evidence might change our conclusions. And right now, the evidence for human-caused climate change is overwhelming. 97% of climate scientists agree. Thousands of studies from multiple independent lines of evidence all point the same direction. The physics is solid. The observations match the predictions. Denying it at this point is like denying that smoking causes cancer or that the Earth orbits the Sun. You can do it, but you're ignoring overwhelming evidence. But here's what gives me hope. Humans are really good at solving problems when we put our minds to it. We've solved problems that seemed impossible. We eradicated smallpox. We sent people to the moon. We invented computers and the internet. We sequenced the human genome. We're editing genes, imaging black holes, detecting gravitational waves. We've built a civilization that, for all its flaws, has lifted billions out of poverty, extended lifespans, reduced violence, increased literacy. Things are far from perfect, but they're better than they've ever been for most people in most places. And we have the knowledge, the technology, the resources to address climate change, to transition to sustainable energy, to protect ecosystems, to build a better future. Whether we do it depends on our choices. Let me tell you about another discovery that changed medicine. Vaccines. The idea seems simple now. Expose someone to a weakened or dead version of a pathogen, and their immune system learns to fight it without getting sick. Then if they encounter the real pathogen, they're protected. But for most of history, this wasn't known. People died from diseases that vaccines now prevent. Edward Jenner pioneered vaccination in 1796 by inoculating a boy with cowpox and showing it protected against smallpox. But the mechanism wasn't understood. Over the next century, vaccines were developed for rabies, typhoid, cholera, plague, and other diseases, mostly through trial and error. In the 20th century, as we learned more about the immune system, vaccine development became more sophisticated. We developed vaccines for polio, measles, mumps, rubella, tetanus, diphtheria, pertussis, and dozens of other diseases. We saved millions of lives. Children who once died routinely from measles or whooping cough now grow up healthy. Polio, which paralyzed hundreds of thousands every year, has been nearly eradicated. And smallpox, which killed hundreds of millions throughout history, has been completely eradicated. The last natural case was in 1977. The disease no longer exists in the wild. That's an unprecedented achievement. We took a disease that had plagued humanity for millennia and made it extinct. And then came COVID-19, a pandemic that killed millions and disrupted the entire world. But we developed vaccines in record time. The first COVID vaccines were authorized less than a year after the virus was sequenced, the fastest vaccine development in history. And they used a new technology, mRNA vaccines. Instead of injecting weakened virus, we inject genetic instructions wrapped in lipid nanoparticles. Our cells use these instructions to make a piece of the virus, the spike protein. Our immune system recognizes this protein as foreign and develops immunity. This approach has advantages. It's faster to develop, easier to adapt to new variants, and potentially safer. And now that we've proven it works for COVID, we're developing mRNA vaccines for flu, for HIV, for cancer. The technology that emerged from a pandemic might revolutionize medicine. But vaccines only work if people take them. And we're seeing rising vaccine hesitancy, driven by misinformation, distrust, fear. People refusing vaccines for themselves and their children. Outbreaks of measles and whooping cough in communities with low vaccination rates. Children dying from preventable diseases. It's heartbreaking. It's frustrating. And it's a reminder that scientific knowledge alone isn't enough. We also need education, communication, trust, willingness to accept expertise. Which brings me to the printing press. Johannes Gutenberg's printing press in 1450 made books affordable and widely available for the first time in history. Before the printing press, books were hand-copied, expensive, rare. Most people couldn't read, and even those who could had limited access to books. The printing press changed that. Books became cheaper. Literacy rates increased. Knowledge spread. Ideas spread. The Reformation, the Scientific Revolution, the Enlightenment, all were enabled by the printing press. It democratized information. And now we have the internet, which is doing the same thing again but faster and more thoroughly. Any information you want is available instantly. You can learn anything, connect with anyone, share ideas globally. But just as the printing press enabled both the spread of knowledge and the spread of propaganda, so does the internet. Misinformation spreads as easily as truth, maybe easier. Conspiracy theories flourish. Echo chambers form where people only hear opinions they already agree with. Algorithms optimize for engagement, which often means outrage and polarization. We have access to all human knowledge, and we use it to argue with strangers and watch cat videos. It's simultaneously wonderful and terrible. So how do we navigate this? How do we distinguish truth from falsehood, good information from misinformation, expertise from nonsense? Science gives us a method: make observations, form hypotheses, test predictions, update beliefs based on evidence. It's not perfect. Scientists make mistakes. Studies are sometimes wrong. Fraud occasionally happens. But over time, through replication, peer review, building on previous work, the truth emerges. We correct our mistakes. We refine our understanding. We get closer to reality. But science also requires humility, recognizing that we don't have all the answers, that our current understanding is probably incomplete or partly wrong, that new evidence might change our minds. The greatest scientists are those who say, "I don't know," when they don't know, who admit mistakes, who change their views when evidence demands it. Certainty is the enemy of science. Curiosity is its lifeblood. Let me tell you about one more discovery: neuroplasticity. For a long time, scientists believed that the adult brain was fixed, unchangeable. Brain cells don't divide, so once you reach adulthood, the brain you have is the brain you're stuck with. Damage was permanent. Learning was limited. But this turned out to be wrong. The brain is plastic, constantly changing, rewiring itself based on experience. When you learn something new, your brain forms new connections between neurons. When you practice a skill, the relevant brain regions grow. When you stop using an area, it shrinks. Stroke patients can recover lost functions as other brain regions take over. Blind people's visual cortex gets repurposed for hearing and touch, making them more sensitive in those senses. The brain is incredibly adaptable. This has practical implications. It means you can learn new things at any age. It means recovery from brain injury is possible. It means mental practice, visualizing movements without physically doing them, actually changes your brain and can improve performance. It means meditation and mindfulness can physically alter brain structure, increasing gray matter in areas associated with attention and emotional regulation. Your experiences shape your brain. Your choices matter. But it also means trauma can change your brain. Chronic stress, abuse, neglect, especially in childhood, can alter brain development in ways that increase risk of mental illness, addiction, and physical health problems. Understanding this changes how we think about these issues. They're not moral failings or personal weaknesses. They're the result of brain changes caused by experiences. And if experiences can change the brain for the worse, they can also change it for the better. Therapy, supportive relationships, new positive experiences can help the brain heal and develop new, healthier patterns. This understanding is transforming mental health treatment. We're moving from a purely chemical model, where mental illness is just a matter of neurotransmitter imbalances fixed with drugs, to a more holistic model that includes therapy, lifestyle changes, social support, and yes, sometimes medication. We're recognizing that mental health is as important as physical health, that it's okay to not be okay, that seeking help is a sign of strength, not weakness. And we're developing new treatments. Deep brain stimulation for depression. Ketamine for treatment-resistant depression. Psychedelic-assisted therapy for PTSD and addiction. Transcranial magnetic stimulation. Optogenetics, using light to control genetically modified neurons. Brain-computer interfaces that let paralyzed people control robotic limbs with their thoughts. We're at the dawn of being able to directly interface with the brain to read neural activity and write information back in. The implications are staggering. It could treat neurological and psychiatric disorders. It could restore lost senses or functions. It could enhance cognitive abilities. It could let us communicate telepathically, share experiences directly brain to brain, upload and download memories and skills. This sounds like science fiction, and maybe it is. Maybe these technologies won't work, or won't work the way we hope, or will have consequences we don't foresee. But consider how much has changed in the last century, the last 50 years, the last 20 years. We went from the first powered flight to landing on the moon in 66 years. We went from the first programmable computer to smartphones in everybody's pockets in 70 years. We went from discovering DNA structure to editing genes with CRISPR in 60 years. The pace of change is accelerating. What seems impossible today might be routine tomorrow. But with great power comes great responsibility. Every discovery that gives us new capabilities also gives us new ways to cause harm. Nuclear energy can power cities or destroy them. Genetic engineering can cure diseases or create bioweapons. Artificial intelligence can solve problems or spread misinformation and automate warfare. We need wisdom to match our knowledge, ethics to guide our technology, foresight to anticipate consequences. And that brings me to the final question I want to leave you with. What's the next great discovery? What breakthrough waiting to happen will transform our world as profoundly as gravity, evolution, DNA, penicillin, quantum mechanics, or CRISPR? Is it fusion energy, solving our energy and climate problems? Is it quantum computing, revolutionizing computation itself? Is it artificial general intelligence, creating minds that match or exceed human intelligence? Is it life extension or rejuvenation, dramatically increasing human health span and lifespan? Is it contact with extraterrestrial intelligence, revealing we're not alone in the universe? Is it a theory of everything, unifying quantum mechanics and gravity, explaining the fundamental nature of reality? Is it something we haven't even imagined yet, that will seem obvious in hindsight but that no one is currently pursuing? We don't know. We can't know. That's what makes it a discovery. But here's what we do know. Someone, somewhere right now, is working on something that will change everything. Maybe they know it. Probably they don't. Maybe they're a famous scientist at a top university. Maybe they're a graduate student whose advisor doesn't think their project will amount to anything. Maybe they're an amateur working in their garage. Maybe they're not even trying to make a discovery, but will stumble onto something by accident, like Fleming with penicillin or Penzias and Wilson with the cosmic microwave background. And maybe, just maybe, it's you. Maybe you'll be the one to ask the question no one's asked, to notice the pattern everyone else missed, to connect ideas that seemed unrelated, to pursue the experiment everyone said wouldn't work. The people who made the great discoveries weren't superhuman. They were curious, persistent, willing to fail, open to being surprised. They asked questions and followed where the answers led, even when it took them somewhere unexpected. They built on the work of those who came before, standing on the shoulders of giants. And they changed the world. Because here's the beautiful, terrifying, awe-inspiring truth. We live in a universe that can be understood. It operates according to laws, patterns, principles we can discover. Every mystery we've solved has revealed deeper mysteries beneath. Every answer has led to new questions. And there's no end in sight. We're not close to understanding everything. We've barely begun. There are frontiers in every direction. Outward to the cosmos, inward to the quantum realm, forward in time to the future of life and mind, backward to the origin of the universe, sideways to the nature of consciousness and reality itself. We are surrounded by wonders. The fact that you're alive, that you can read these words and understand them, that your brain, made of atoms forged in dying stars, can contemplate its own existence, is itself a miracle made possible by billions of years of cosmic evolution. You are made of star stuff, as Carl Sagan said. You are the universe becoming aware of itself. And everything you know, every technology you use, every year you're likely to live beyond what your ancestors managed, every comfort and convenience and capability, exists because someone asked a question and wouldn't stop until they found an answer. The greatest scientific breakthroughs in human history aren't just names and dates and facts to memorize. They're stories of human curiosity and persistence and ingenuity. They're reminders that we're capable of far more than we imagine. They're invitations to wonder, to question, to explore. They're the foundation we build on, each discovery making the next possible. And they're ongoing. The story isn't over. The next chapter is being written right now. And who knows? Maybe you'll be part of it. Maybe you'll be the one who discovers something that future generations will marvel at, that will transform how we understand the world and our place in it. Or maybe you'll simply appreciate the wonder of it all, the incredible journey from ignorance to understanding, from darkness to light, from mystery to knowledge. So here's my challenge to you. Stay curious. Ask questions. Don't accept easy answers. When someone says something is impossible, ask why. When you see something that doesn't fit the pattern, investigate instead of dismissing it. When you fail, learn from it. When you succeed, share it. Build on what others have discovered. Stand on the shoulders of giants and see farther than they did. And never forget that every great discovery started with someone wondering about something that everyone else took for granted. The universe is full of secrets waiting to be revealed. The question is, will you be the one to find them?